Hello guys, this is Zoologist AK, and in this video I will talk about types of RNA, in which I will talk about varieties of RNA, but my main focus will be on the messenger RNA, that is mRNA, t-RNA, that is transfer RNA, and rRNA, that is ribosomal RNA. So there are different types of classification of RNA. First, I will talk about classification of RNA based on their function or role in a particular cell or in a particular organism. For example, RNA that is involved in protein synthesis, means during translation process they involve in different types of activity. For example, messenger RNA, as we know that messenger RNA carry information from DNA to ribosome and the form of codon that is a 3 nucleotide base pair or bases, and that code the anticodon in the ribosomal subunit. And second one here is ribosomal RNA that form the small or large subunit of ribosome, and then signal recognition particle RNA, or we call them 7SL RNA. These RNA present in all organism, and this SRP, that is signal recognition particle RNA, has a function that is membrane integration, as a membrane integration molecule. And another ones are transfer RNA, or that transfer amino acid during protein synthesis to the ribosomal subunit. And tmRNA here stand for transfer messenger RNA. It rescues stalled ribosome in case of bacteria during translation process. When ribosome stop moving and it stalled at a position, then this mRNA take its role. So these were the example of RNA that are involved in protein synthesis. Now come to our second point, that is RNA involved in post-transcriptional modification or DNA replication. Here I will talk about different types of example. For example, snRNA, small nuclear RNA. It play a role in splicing process after formation of a pre-RNA from transcription. This pre or nascent RNA underwent a splicing process in which exons are cut down from introns, and a mature RNA product is produced in which exons are aligned to each other. So these small nuclear RNA play a role in splicing in case of eukaryotes and archaea. Then another is small nucleolar RNA. These small nucleolar RNA play a role in nucleotide modification of RNA in case of eukaryote and archaea. Then guide RNA, I also talked in my previous video about this guide RNA. It play a role in mRNA nucleotide modification in case of mitochondria. And another one is RNase P. RNase P has a role in tRNA maturation, in which I will talk that in tRNA maturation, it cut down the part of tRNA, a result in pre-RNA, that is pre-tRNA that formed after transcription, and result in the formation of mature tRNA. It present in all organism. Then RNase MRP. RNase MRP stand for maturation DNA replication. It present in eukaryote, and it has a role in replication of DNA during replication process in case of eukaryotes. And another example here is yRNA. It has a role in RNA processing and DNA replication. It present in all types of animals. And then telomerase RNA component, telomerase RNA component. It has a role in telomere synthesis in most eukaryotes. And another example here is spliced leader RNA. Spliced leader RNA, slRNA, that is spliced leader RNA. It has a function in mRNA transplicing and RNA processing. And next one is regulatory RNA. Regulatory RNA is that regulate the expression of a particular gene inside a cell or a organism. It could be a bacteria and eukaryotes, etc. For example, antisense RNA, as I talked earlier, antisense RNA that bind to mRNA that take information from DNA to ribosome during protein synthesis, and this antisense RNA by using complementary base pairing result in formation of a double strand with the mRNA, and due to which RNase, that is nuclease enzyme, act on this double stranded DNA, in which antisense mRNA, that is a coding RNA, bind together and cleave these double-stranded RNA and result in degradation of messenger RNA. So this antisense RNA play major role in gene regulation, and example of this antisense RNA are aRNA and asRNA. They have major role in like translation attenuation, mRNA degradation, and mRNA stabilization, translation blocking. These RNA present in all organism. Then another example of regulatory RNA are cis-natural antisense transcript. Cis-natural antisense transcript, we could write them like cis-NAT, antisense natural antisense transcript. N stand for natural, A stand for antisense, T stand for transcript. So they play a role in gene regulation. Another example here is CRISPR RNA, or we call them cRNA. They are resistant to parasites by targeting the DNA of that parasite. It could be a virus, or so they target the parasite, the DNA or genome of the parasite that enter in the host organism. It present in bacteria and archaea. Other example of regulatory RNA are microRNA or mRNA. They play major role in gene regulation in case of eukaryotes. And another example here is siRNA, that also play a role in gene regulation in case of eukaryotes. And short hairpin RNA, that is shRNA, short hairpin RNA, also play a role in gene regulation in case of eukaryotes. And other examples are transacting siRNA, or we call them TASI-RNA. They also play in role in gene regulation in case of land plant, not in animals but in land plants. And another example is 7SK RNA. We also write it like 7SK. It is a negative regulating CDK9 and cyclin D complex. It has a function in negatively regulating CDK9 that play a role in cell cycle division and cyclin T complex. And another example here of regulatory RNA is enhancer RNA, that is eRNA, that play a role in gene regulation. So this was all about regulatory RNAs. Now come to our next point, that is parasitic RNA. Parasitic RNA reside as a parasite in a host organism genome. For example, we studied about transposons. Transposons are nothing but jumping genes. They have no role in protein coding or in they are not coding sequence, but they are reside in an organism's genome as a parasite. For example, different examples of parasitic RNA are like retrotransposons. Retrotransposons are self-propagating transposons. They replicate themselves by reverse transcriptase process, through which reverse transcriptase enzyme act on these retrotransposons and result in synthesis of circular DNA molecule, due to which they synthesize DNA from an RNA and replicate themselves. So these are self-propagating and they present in eukaryotes and in some bacteria. Another example of parasitic RNA are viral genome, like double-stranded RNA virus, and that play a role in the genome formation in some case of virus. And positive sense RNA virus negative sense RNA virus, satellite virus, and reverse transcribing virus that form by reverse transcriptase and with the help of reverse transcriptase they form circular DNA and integrate with the host organism genome. So they reside or propagate or integrate and divide or replicate again and again in a host organism and infect that particular organism. So these are examples of parasitic RNA. Now come to another classification of RNA, that is based on coding and non-coding mechanism of RNA. So first is our coding RNA, that is our messenger RNA. As we know that messenger RNA code information from DNA to ribosome during protein synthesis in the form of codons. So another is non-coding RNA. It include ribosomal RNA that form subunits of ribosome, small subunit or large subunit like 5S rRNA, 5.8 ssRNA, and 28 ssRNA. So they form subunit of ribosome in eukaryotes and in bacteria also. So another one is t-RNA, transfer RNA, that transfer amino acids to the ribosomal subunit during protein synthesis. And another example of non-coding RNA are small nuclear RNA. Small nuclear RNA form complex with different types of protein and result in the formation of snRNP complex, small ribonucleoprotein complex, that play a major role during splicing process. And these are different type: U1, U2, U4, U5 and U6, and they result in the formation of splicing complex or splicing machinery and result in the splicing of introns from exon and result in the maturation of an RNA that have exons only. And another example here of non-coding RNA, small nucleolar RNA. These are about 60 to 300 nucleotide base pair long, and these are component of small nuclear ribonucleoprotein and responsible for sequence specific nucleotide modification. Another example here is PV interacting RNA. PV interacting RNA, we called it piRNA. P stand for PV, I stand for interacting, R stand for RNA. These are 24 to 30 nucleotide base pair long, and they maintain genome stability in germline cell. For example, they have a role in gametogenesis during spermatogenesis or oogenesis. And another example of non-coding RNA is microRNA, that is miRNA. These are about 21 to 22 nucleotide long, and they mediate post-transcriptional gene silencing through RNA interference mechanism that we will study during transcription regulation in case of eukaryotes, where an effector complex of messenger interference RNA and enzymes can target a complementary mRNA by blocking the mRNA from being translated and accelerating its degradation. In humans, messenger interference RNA regulate the translation of more than 60% of protein coding genes. So these play a major role in case of regulation of gene expression in case of eukaryotes. And the three main types of RNA that I'm going to discuss in this video in detail, these are transfer RNA, messenger RNA, ribosomal RNA. These are present in both prokaryotes and eukaryotes. We will study them in detail. So first of all, we will talk about messenger RNA, and in messenger RNA we will talk about all these 3 points. And this is our first point: polypeptide chains are specified by ORF. ORF stand here for open reading frame. So let us understand how does these polypeptide chains are specified by open reading frames. As we know that 3 nitrogenous base pair or nucleotides form a codon. For example, AAA, ACA or CAC or ACA, AUG. These are example of codons. So these codons specify a particular amino acid, and these amino acids or codons reside in a open reading frame. So let us understand what are these open reading frame. These open reading frame are nothing but the protein coding region that are present inside a mRNA and composed on non-contiguous string of codon. Here non-contiguous string means they are aligned to each other on a chain of mRNA and are not in a overlapping state. So each open reading frame specify a single protein and that a single protein and consists a start and at internal site within an mRNA means they consist a start codon and a stop codon that and the mRNA chain inside this open reading frame. And these open reading frame read in a particular frame that the frame that in which these are written is set by first codon, that is our start codon. Why we call it start codon? Because this is open for translation until the last codon is reached, and last codon will be the stop codon. This picture here showed us the 3 reading frame of E. coli trip leader sequence. This shows trip leader sequence and it has 3 reading frames, first, second and third. Here this codon at 5 prime region showed us the start codon and this shows us the stop codon. Let us understand about start codon and stop codon. Start codon, it start means the reading frame is read from the start codon that is present at 5 prime end of open reading frame and proceed one codon at a time. As we can see, one codon at a time up to which it reaches up to the 3 prime end that consists a stop codon UGA. So in case of bacteria, start codon could be either 5 prime AUZ to 3 prime, or it could be GUZ, or it could be UUG. But usually this AUZ is present as a start codon in case of bacteria, but these are also present sometime in case of bacteria. But as we talk about eukaryotes, then there is the start codon, that is our AUZ. This start codon has some function. First, they specify the first amino acid that is to be incorporated into a growing polypeptide chain. As we can see here, this M is showed here that AUZ encode for amino acid that is methionine, and second function of the start codon is that it defines the reading frame for all subsequent codons, means reading start from here and adds here. So it defines the reading frame for all subsequent codon that lie after the start codon. Because codons are 3 nucleotide long and any stretch of mRNA could be translated into 3 different reading frames. So here these 3 reading frames are shown for E. coli trip leader sequence that could be translated into a 3 reading frames. By setting the location of first codon, the start codon determine the location of all the subsequent codons. And stop codon could be UZ, UAZ, UZA or UAA. It defines the end of open reading frame and signal the termination of a polypeptide. So open reading frames are contiguous stretch, means they are not overlapping. As you can see here in the picture, they are aligned to each other and they could be read in a particular frame that is start from start codon and at the stop codon. So these reading is set by first codon that is open to translation until the last codon is reached in case of open reading frame. So mRNA has at least one open reading frame, and the number of open reading frames per mRNA is different between eukaryotes and prokaryotes. In case of prokaryotes, there could be two or more open reading frame that encode multiple polypeptide chain, and mRNA that contains multiple open reading frames known as polycistronic mRNA. And this is the example that is shown in the picture is of polycistronic mRNA. And a mRNA chain that is present in case of eukaryotes encode single open reading frame and consist or called as or known as monocistronic mRNA. Here, as we can see, these different codons that are subsequent to the start codon code for different amino acid. For example, AUG encode for M, that is stand for methionine, and AAG encode for K, that is lysine, and GCA encode for A, that is alanine, and AUU encode for I, that is our isoleucine, and UUC that encode for F, that is phenylalanine, and GUA that encode for V, that is valine, and CUZ that encode for L, that is our leucine, and here K stand for lysine, G stand for glycine, and W stand for tryptophan, and here again it's UZZ encode for tryptophan, and R stand for arginine, and T stand for threonine, and S stand for serine. And then come to our stop codon that will stop the translation process. So this was how does this polypeptide chain is specified by open reading frame. Now come to our second point, that is prokaryotic mRNA have a ribosome binding site that recruit the translation machinery. So let us understand about this RBS, that stand for ribosome binding site, or how does this recruit a translation machinery to the prokaryotic mRNA. As we know that for translation to start, ribosome must be recruited or bind to the mRNA chain, and where it will bind to the RBS, that is shown in the mRNA chain, and it is present upstream. As we can see here, this is start codon, or this is the sequence. Here we can see AUG is the start codon, and this green color sequence GGA GGE, this showed a Sil-Dargano sequence that consists a ribosomal binding site, that is shown here RBS. So this RBS is nothing but 3 to 9 base pair long stretch sequence that is present on 5' end of mRNA, and it present upstream of this start codon. And this ribosomal binding site consists a sequence that is shown in this green color GGA GG. It is complementary to the 3' end of 16S ribosomal subunit. As we know that in case of bacteria, 3 types of ribosome rRNA are present: 23S rRNA, 5S rRNA, or 16S rRNA. 23 and 5S rRNA form the component of large subunit of ribosome, and 16S rRNA in case of prokaryotes form the component of small subunit. So this 16S rRNA present in the small subunit of ribosome, and it consists a sequence at the 3' end that is complementary to the sequence that is GGA GG that is present in the ribosomal binding site. This ribosomal binding site is present upstream to the start codon, and it is a 3 to 9 base pair long and present on the 5' end of the mRNA chain. So this RBS, that is ribosomal binding site, base pair with this RNA, that is 16S rRNA, and align the ribosome with the beginning of this open reading frame or at the start codon, and core of this region of the 16S RNA has a sequence that we know that CCU. And prokaryotic ribosomal subunit consists a sequence that could be 3 to 9 base pair long, and it will be AGG, AGZ, AGG. As we can see here, GGA, GG. So here this A is missing, and it could be AZZ, AZZ. As we know that this is the particular sequence AGG, but in different mRNA chain it could vary from 1 or 2 base pair. Here, this picture below shows the monosysteric mRNA that consists a open reading frame, and it is present in case of eukaryote. Here, a question arises that, or a important point arises, that the extent of complementarity between these sequence that is present in 16S rRNA that form the small subunit of ribosome and the ribosomal binding subunit that consists a sequence GGA, GGA, GZA. So these sequence form the complementarity, and there is the proper spacing between start codon and this ribosomal binding subunit. It actively regulate the translation process, and if there is the high complementarity between the sequence and proper sequencing between the start codon and RBS, then active translation will occur. And if there will be the limited complementarity or poor spacing between the start codon and RBS, then the lower level of translation will occur. In some prokaryotes, open reading frame lack a strong RBS. For example, this is our mRNA chain that consists of open reading frame that lack a strong RBS, and this RBS is less strong or unable to bind the ribosome to its sequence, that is Dalgarno sequence. And here will be the start codon AUZ, and here will be the stop codon UZA. It will lie 5 prime to 3 prime, and this open reading frame that consists this less strong ribosomal binding site that is unable to bind to ribosome, this will not present as a first open reading frame, rather than it will lie in or after another open reading frame in a polycistronic message. As we know that prokaryotic mRNA consists more than one ORF, and they could bind more than one ribosome at a time, so they are called polycistronic mRNA. And in this case, here will be another open reading frame that will consist here UZA, that is the stop codon, and here will be the AUZ. So in this case, when this less strong ribosomal binding site or open reading frame that is unable to bind to ribosome lie after the another open reading frame that is present here, in this case the start codon of the downstream open reading frame, that is AUZ, that is present here, will be overlap the 3 end of the upstream stop codon, that is UZA, that lies here. So it will form a sequence 5 prime AUZA. It will consist AUG, that is start codon, and UZA, that is stop codon. So it consists start and stop codon. Thus a ribosome that has just completed translating the upstream open reading frame is positioned to begin translating from the start codon for the downstream open reading frame, and this arrangement is for the recruitment of ribosome. This link translation is called coupling translation. And in this translation of the downstream open reading frame require translation of the upstream open reading frame, that is why we called it coupling translation. So if there is a mutation that lead to a premature stop codon in the upstream open reading frame, if upstream open reading frame consists a mutation due to which it lead a premature stop codon in the upstream, and then it will prevent the translation of this downstream open reading frame too, because this occur in a coupling translation mechanism. Now come to our 3 point, that is eukaryotic mRNA are modified at their 5 or 3 prime end. 5 prime end that occur cap there occur 5 prime capping that is located at extreme 5 prime end, and it is a methylated guanine nucleotide that is joined to the 5 prime end of the mRNA by an unusual 5 prime to 5 prime linkage. How does it occur? As I talked before in my videos, that the incoming guanine consists three phosphate group, that is alpha, beta and gamma, where one phosphate is left, and the ribose subunit that is present in mRNA chain will lose its gamma phosphate, and then there will be the linkage occur at 5 prime to 5 prime by their 3 phosphate group. So this resulting 5 capping at 5 prime end of the RNA is required to recruit the ribosome to mRNA chain in case of eukaryote. Once this ribosome is bound to the ribosomal binding site, that is RBS, it will move in 5 prime to 3 prime direction, up to which it does not encounter the start codon, that is our AUG. So this process is called as scanning. And poly A tail enhance that occur at 3 prime end, where about 200 nucleotide base pair of adenine will bind to 3 prime end, and this will enhance the level of translation of mRNA by enhancing the recruitment of translation initiation factor. So these are the modification that facilitate translation in case of eukaryote. And other feature of eukaryotic mRNA that stimulate translation process is a purine 3 bases upstream of start codon, and a guanine immediately downstream, and this will create a sequence that is called 5 prime G A N A U G G 3 prime, and this sequence is identified by Marilyn Kojak. That is why it is referred to as Kojak sequence. Many eukaryotic mRNA lack this sequence, but this increase the efficiency of translation in case of eukaryotes. So these were the modification that facilitate translation in case of eukaryote. Come to another RNA, that is transfer RNA, and in this topic I will talk about this point, that will discuss the structure of RNA, and this point to discuss the role of this tRNA. So let's discuss our first point, that is transfer RNA are adapters between codons and amino acids. Transfer RNA are adaptive molecules. As we know that transfer RNA adapt or bind a codon or multiple codons and transfer them to the ribosomal subunit during translation process. And these tRNA, that is transfer RNA, are multiple types, but each is attached to a specific amino acid. And these tRNA about 75 to 95 ribonucleotide long in length, but these sequence of these tRNA varies. But some sequences are common among the different tRNAs that we called conserved sequence, and this conserved sequence is present in all tRNA at the 3 terminal end or at 3 terminus. This sequence is 5 prime CCE 3 prime, and this is the site that is attached to the cognate amino acid that it will transfer to the ribosomal subunit. And another similarity between different types of tRNA is the presence of several unusual bases in their primary structure, and these are created by post-transcriptional machinery by enzymatic modification of normal base in polynucleotide chain. For example, here you can see pseudouridine. This is formed by isomerization reaction in which the site of attachment of uracil to the sugar is changed. As you can see here, ribose is attached to first nitrogen in case of uridine, but in pseudouridine it is attached to 5th carbon. And these just like pseudouridine, there are some another types of modification of normal bases like dihydrouridine. It is derived from uridine by enzymatic reduction of double bond. As you can see here, in 5th and 6th position in uridine there is a double bond double bond is present. But in case of hydroxy means OH, but here dihydro, because two hydrogen atoms are get attached at fifth carbon and sixth carbon, due to which this double bond is converted into single bond. So this uridine result in formation of dihydrouridine by base modification process after post transcriptional modification. And other unusual bases that is found in tRNA molecule are hypoxanthine, thymine. As we know that usually uracil is present, and if there will be the thymine, then it is produced by normal base pair modification. And methyl guanine. These modified base lead to improved tRNA function. For example, hypoxanthine play an important role in the process of carbon recognition by certain tRNA molecules. Now come to our second point, that is transfer RNA. Here this is tRNAs. So transfer RNA share a common secondary structure that resembles a clover leaf structure. As you can see here, this is a clover leaf structure. And what are the features of this tRNA clover leaf are: first is acceptor arm or acceptor stem. This acceptor stem, it is the site that attached to the incoming amino acid, and it is formed by pairing between the 5' to 3' CCA base pair that is present at the extreme terminal end of this acceptor arm. And another feature of this clover leaf is pseudouridine loop, that is due to the presence of unusual base, that is pseudouridine, and that is formed by modification of normal base, and it is normally found in the sequence 5' thymine pseudouridine CG and 3'. This is found in the sequence 5' thymine pseudouridine CG and 3'. And another feature is the presence of D loop, and this D loop is characterized by presence of dihydrouridine. As we know that here this dihydrouridine is formed by replacing this double bond into single bond by binding of two hydrogen at the position of fifth carbon and sixth carbon. So this D loop is formed due to the presence of dihydrouridine in this loop structure of mRNA, that is formed by complementary base pairing. Here this red color shows complementary base pair between or within the chain of transfer RNA that result in the formation of clover leaf like structure. And another feature is the presence of anticodon. Anticodon loop contain a anticodon. And what is the feature of this anticodon? Here will be the presence of anticodon and a mRNA chain that will consist a codon will bind by complementary base pairing to this anticodon and result in the translation. Here this the features of anticodon is that 3 nucleotide long sequence. It will be the 3 nucleotide long sequence, or it will consist 3 nucleotide bases, and it is responsible for recognizing the codon that will base pair with and result in base pairing with the mRNA. The anticodon is always bracket on 3' end by a purine base. Purine could be adenine and guanine. So at 3' end, here will be the adenine or guanine nucleotide base, and at 5' end uracil base will be present, that result in the formation of anticodon in this anticodon loop. And next feature of this clover leaf is variable loop. Why we call it is a variable loop? Because it varies in size up from 3 base pair to 21 base pair sequence, and it is a site that reside between anticodon loop and the pseudouridine loop. And our third point is transfer RNA have an L-shaped three-dimensional structure. As we know that clover leaf reveal region of self-complementary or complementary sequence within the tRNA. As we can see here, these red dots show here complementary base pairing. And X-ray crystallography reveals a L-shaped three-dimensional structure that is shown in here of tRNA, in which the terminus of acceptor arm, as we can see here, this is our acceptor arm. So the L-shaped structure showed us that the terminus of acceptor stem is at one end of the molecule, and the anticodon stem loop is 70 angstrom away at the other end. As we can show here is our anticodon, and in this structure here this anticodon is 70 angstrom away from this acceptor arm. As we can see here, the acceptor stem that is present in here, this acceptor arm or stem, and pseudo-uridine loop form this yellow region showed here, pseudo-uridine loop. This pseudo-uridine loop form and extended helix in the final tRNA structure. As we can see here, this form a extended helical structure. Similarly, the anticodon loop that is shown in dark blue color and stem of D loop that is shown in this here form a second extended helix, as we can see here in this X-ray crystallography diagram that is shown in here. These two extended helix align at a right angle. As we can see, this formed a straight line and this also formed a straight line. So these align at a right angle form or right angle configuration with the D loop and with the D loop that is here, and the pseudo sigma subunit that is in yellow color, and these result in the coming together of this D loop structure and this pseudo uridine loop structure. And there are three kinds of interactions that stabilize these L-shaped structure. First is the formation of two extended regions. One is here and second is here. So these formation of two extended region of base pair resulting in base stacking interaction that is similar to double stranded DNA that stabilize this L-shaped structure. Second is hydrogen bond between bases in different helical region that are brought near each other in a 3D space by tertiary structure. And third one is base interaction that is result in unconventional base pairing. That is unconventional means here non-Boston Crick base pairing. So these three features stabilize this kind of interaction that result in formation of L-shaped structure. Well, let's see our first point in the role of tRNA, that is charging of tRNA. Let us understand this. As we can see here, this is the acceptor arm and here adenosine is present. Amino acid will bind to this adenosine second or third hydroxyl group that is present on second carbon or third carbon. This amino acid will form a acyl linkage with this adenosine. How does this acyl linkage will form? Amino acid will consist a carboxylic group that will bond with this hydroxyl group and result in the formation of a acyl linkage that is high energy bond. Why does it high energy bond? Because its hydrolysis result in a large change in free energy. So when this acyl bond is broken down, a large amount of energy is released that is utilized to the formation of peptide bond during polypeptide chain formation. Our second point, in the amino acid tRNA synthase that charts the transfer RNA in two steps. First step that is shown in here is adenylation reaction. Here amino acid react with ATP molecule in the presence of an enzyme that is the pyrophosphatase, due to which pyrophosphate will release, and this carboxyl group will result in the formation of a bond with this AMP moiety, that is uh, adenosine monophosphate, that will result when this pyrophosphate will release out of this ATP molecule, and this process is called as adenylation. So here amino acid attached to adenylic acid via high energy bond, that is an ester bond, that is formed when this carbonyl group attached to AMP moiety. And second reaction is the charging of tRNA. It results when this amino acid attached with the 3 hydroxyl group that is present on adenosine, that is present on 3' end of acceptor arm of tRNA, and this is result in formation of a AMP moiety from this amino acid, and it is caused by an enzyme that is synthetase. Synthetase are two types, synthetase first and synthetase second. Synthetase first bind this amino acid to 2 hydroxyl group, and synthetase 2 bind this amino acid to 3 hydroxyl group, and this transfer of amino acid is result in the charging of tRNA and result in the removal of AMP moiety, that is adenosine monophosphate. Now come to our next point, that is each amino acid tRNA synthase attaches a single amino acid to one or more tRNA. So let us understand this. On tRNA synthesis can recognize and charge more than one tRNA. So one and only one tRNA synthesis attaches each amino acid to all of the appropriate tRNA. Most organisms have about 20 different tRNA for 20 amino acid, but this is not always the case that is found in different organisms. For example, some bacteria lack a synthetase for charging the tRNA for glutamine with its cognate amino acid, and a single species of amino cell tRNA synthetase charges tRNA glutamine as well as tRNA glutamate. So that is glutamate tRNA glutamine is animated to glutamine tRNA glutamine by an enzyme. Here the prefix identified the attached amino acid. So the presence of second enzyme that animate a glutamate to glutamine and result in the identification of the tRNA that code for glutamate. This presence of second enzyme removes the need for a glutamine tRNA synthetase, because on amino acid tRNA synthetase can never attach more than one kind of amino acid to a given tRNA. Next point is the tRNA synthetase recognize unique structure features of cognate tRNA. As we know that an amino acid tRNA synthetase can never attach more than one kind of amino acid to a given tRNA. So there need to be a structure feature to recognize a specific amino acid to bind to this tRNA molecule by tRNA synthetase. Let us understand the mechanism. How does this tRNA synthetase recognize the specific tRNA that will bind to a specific amino acid? This is the structure that is shown here that represent the structure feature that play a role in the specificity recognition. Here the specificity determinant are clustered at two distant sites on the molecule. First is acceptor stem or acceptor site, and second is anticodon site or anticodon loop. So this acceptor stem if there is a change of a single base pair in the acceptor stem, then that change in one base pair result in a discriminator base pair that will present in this acceptor stem, and it is sufficient to convert the recognition specificity of a particular tRNA molecule from one synthetase to another. And what is the role of this anti-codon loop? It contributes to discriminator. It is used for recognition by tRNA synthetase because it is the defining feature of a tRNA. Because as we know that this anticodon code for codon that are present on mRNA during translation machinery or during protein synthesis. So it consists an anticodon that dictate the amino acid sequence that the tRNA is responsible for incorporating into a growing polypeptide chain with the help of mRNA codon. But each amino acid is usually specified by more than one codon. So the recognition of this anticodon cannot be used in many cases. For example, if we take an example of serine that have a variety of different anticodon, or serine is specified by 6 codon, that including base pair like 5 prime to A, G, C 3 prime or 5 prime U, C, A 3 prime. These are completely different sequence A, G, C and U, C, A. So different codon specify the serine amino acid and they are different in sequence. So this tRNA for serine have a variety of different anticodon to code these different codons, as we know that 6 different codon code for a amino acid that is serine. So there will be the different anticodon present on this anticodon loop that is present in the structure of transfer RNA. So to recognize this different tRNA is not possible by a single synthetase. So to recognize its tRNA synthetase for serine rely on the determinant that is present outside of this anticodon loop. So here the determination or specificity factor is determined by this determinator that is present outside of the anticodon region that lie on transfer RNA. Now come to our next one, that is amino acyl tRNA formation is very accurate. Let us understand this with an example. If amino acid, as we know, amino acid tryptophan and cysteine that differ in size, shape and a chemical group, but there is less than 1 in 1000 transfer RNA is charged with incorrect amino acid. Similar example is of amino acid tyrosine and phenylalanine that are very similar looking amino acid, but one difference is of hydroxyl group that is present in tyrosine and absent in phenylalanine. So the opportunity for forming a strong and energetically favorable hydrogen bond with hydroxyl moiety of the tyrosine but not of the phenylalanine allows the synthetase for tyrosine, that is tyrosyl tRNA synthetase that discriminate tyrosine from phenylalanine. Here the enzyme that act is tyrosyl tRNA synthetase. Another case is of valine and isoleucine that differ by single methyl group that is present in on isoleucine. Here enzyme that act is valine tRNA synthetase that can strictly exclude this isoleucine from its catalytic pocket because isoleucine is very large than valine. But if the concentration of this valine and isoleucine is equal, then isoleucyl tRNA synthetase will bind to isoleucine because it consists an extra methyl group. So it will produce an extra 2 to 3 kilocalorie per mole of free energy. So it will bind to isoleucine 100 fold more likely than binding to this valine amino acid. So before understanding our second point, let us understand this point. As we get to know that these isoleucine transfer RNA bind to isoleucine 100-fold more likely than binding to valine. Thus, valine could be attached to isoleucine tRNA about 1% of time. This is inacceptable high error, but the actual frequency of miscorporation that we studied is 1 in 1000 transfer RNA that is charged with a incorrect amino acid. Then how does this additional level of fidelity is achieved? Let us understand this in our next point, that is some amino cell tRNA synthetase use an editing pocket to charge tRNA with high accuracy. Let us understand this point with an example. Let us take an example of a synthetase that is isoleucine tRNA synthetase. It consists two site. One is catalytic pocket that will function of or that will conduct adenylation reaction, and second is editing pocket. Or this editing pocket is a deep cleft in the enzyme that has a role in proofreading mechanism that protect or produce the adenylation reaction. So AMP, adenosine monophosphate valine, as well as adenylation of other amino acids such as aniline can fit into this editing pocket, where it is hydrolyzed and released as free valine and free AMP molecule. In contrast, AMP isoleucine is too large to enter this editing pocket. That is present in isoleucine transfer RNA synthetase enzyme. Therefore, it is not subject to hydrolysis. As a consequence, this isoleucine transfer tRNA synthetase discriminate against valine twice in the initial binding and adenylation of the amino acid, and discriminating by a factor of about 100. And then in the editing of adenylated amino acid, again discriminate by a factor that is 100. As I told that these will discriminate twice for an overall specificity of 10,000 fold. That is, it will consist or create an error of about 0.01%. So we can say that this editing pocket is used by amino acid tRNA synthetase to charge tRNA with high accuracy. To our last point in the role of transfer RNA, that is ribosome is unable to discriminate between correctly and incorrectly charged tRNA. As we know that ribosome cannot distinguish between correct and incorrect charged tRNA. This point is supported by two experiments. One is genetic and second is biochemical. In case of genetic experiment, it involves isolation of mutant tRNA that carries a nucleotide substitution in anticodon or in anticodon loop. Here, subset of tRNA can be mutated in their anticodon but still be charged with their usual cognate amino acid. Due to this, a consequence arises, that is, the mutant transfer RNA deliver its amino acid to the wrong codon. This is checked by auxiliary protein that bind the correct charged transfer RNA to the ribosome. And another mechanism is biochemical mechanism or biochemical experiment. Here it illustrates the point that ribosome recognize transfer RNA, not the amino acid that is it is carrying or carried by transfer RNA. So ribosome only recognize transfer RNA that is charged, and charged transfer RNA cysteine And for example, take a example here of charged transfer RNA that is charged with cysteine amino acid, or we can say here this RNA tRNA cysteine. Here, this prefix identified the amino acid and superscript identified the nature of transfer RNA. And this cysteine can be converted to alanine by a chemical reduction reaction. And when added to a cell-free protein synthesizing system, alanine tRNA cysteine introduce alanine at the codon and specify insertion of cysteine. So translation machinery rely on the high fidelity of amino acid tRNA synthetase to bind the correctly charged tRNA to the ribosomal subunit. Now come to our last RNA, that is rRNA or ribosomal RNA. We also called it ribosomal ribonucleic acid. Ribosomal ribonucleic acid. It is a type of non-coding RNA and it is the primary component of ribosome that is an essential to all cell. This rRNA is act as a ribozyme, that is act as an enzyme during protein synthesis, and this ribosomal RNA is transcribed from ribosomal DNA and then bound to ribosomal protein to form small and large ribosomal subunit. This is the physical and mechanical actor of ribosome that force transfer RNA and messenger RNA to process and translate the latter into protein. It is the prominent form of RNA that is found in most cells. About 80% of cellular RNA is formed by ribosomal RNA. There are two types of RNA found in prokaryotic ribosome, that is 20S, 5S in the large subunit, and in small subunit is 16S ribosomal RNA. In eukaryotes, for example, in case of yeast, the large subunit of RNA consists 5.8 sRNA, 5 sRNA and 28 sRNA. But in small subunit of ribosome, only 18S ribosomal RNA are present, and we will study this ribosomal RNA in detail when we will study about ribosome before studying the translation process in detail. So this was all about varieties of RNA, and if you like my video then please thumbs up, and if you have any query then please write in the description box given below, and please support me by subscribing my channel. Thank you so much for watching my videos.